The hope tonight is that we're going to have a conversation, starting with the questions you might have, and then over dinner, about what I can say with great confidence is the most transformative technological advance that I've seen. And so there's almost no question, I think, in people who think about this issue's mind, that the changes in DNA sequencing are going to have an accelerating impact on all of us. And so what encapsulates this is the fact that we're going to be able to, in short order, read out the blueprint for each of us for $1,000 each. Deciphering our DNA in this manner will lead to an area of precision medicine, and this is certainly going to affect all of us, but it's going to have a particular impact on our children, for whose future this data will be a critical piece. Now, the story of DNA, some of you know already, begins here at Rockefeller University. In 1944, to everyone's amazement, Avery, McCarty, and McLeod determined that the genetic material, the blueprint that's carried on from generation to generation, from parent to child, from species to species member over the eons, is DNA. I mean, this was at the time astounding. DNA is, in some senses, a simple material, and people could not imagine that something with as, at least on the surface, simple a structure as DNA could be the genetic material. But indeed it was, and this ushered in changes in biomedical science that are really continue to grow and astonish everyone who's part of it. Now, at the time, the realization that DNA was the genetic material, as shown by this group of investigators, led a colleague of theirs, Rollin Hotchkiss, to write a poem that he entitled Revolution or Evolution, because DNA is the substrate on which evolution takes place. And he wrote, "Once in a prehistoric blister in the prehistoric slime, I thought I'd like to have, the slime thought I'd like to have a sister, and to save himself some time, he wrote down the mystic secrets of his protoplasmic virus on some stuff that he found handy, a sort of primitive papyrus." A primitive papyrus here was DNA. A billion years later emerges our species, the products of a billion years of evolution. Here we are. And we now have the ability to understand in ever greater detail why these two typical humans have the characteristics they do. The starting point for this was the deciphering of the human genome. I'll tell you more about this in a moment, in 2003. And the pace at which discoveries emanating from this are being realized is accelerating. Now, a brief review. DNA is carried on chromosomes. And as I mentioned, DNA is the blueprint for the production of all the critical components of our body, largely protein. Now, DNA is a messenger, as it were. It carries, as I said, a blueprint. It's copied into RNA, and this RNA is read out into protein. And proteins are sort of the business end of this information scheme. There are thousands, tens of thousands of proteins in our body, and they determine, to a large extent, why we're tall, what color eyes we have, and they perform every function either directly or indirectly. Now, the way this information flow works is as follows. DNA is composed of a simple structure of only 4 what are called bases, and these can be viewed as being letters. There are 4 letters read out in a sequence that extends for billions of units. And each of these bases, 3 at a time, read out here, GCA and so on and so forth, are read out into amino acids, which are the building blocks of proteins. So what's written here is the array of these 4 letters. It can be G, C, A, or T at any one position. That's illustrated here. And as this gets read out, when the cellular machinery sees a GCA, it puts the amino acid alanine in place, reads AGA, puts arginine in place, and so on and so forth. And so the DNA is a blueprint that's read out into proteins, and proteins do most of the business of our bodies. Actually, this code has been well established now, and what you'll see here is the first letter, the second letter, and the third letter, and each of these triplets is read out with a different amino acid And this allows proteins to be synthesized using the DNA blueprint. Now, this entire blueprint, as I've been alluding to, can be read out for ever lower cost. Some of you may have had your genome sequenced, your blueprint sequenced, as it were, by 23andMe, a screenshot from which is shown here. Who has had their DNA read out by 23andMe? Who was happy about it? We'll talk about this. Well, some things emerge from genome sequence of the sort that 23andMe can provide, and that was first preferred in 2003 with the draft genome sequence. First of all, humans have surprisingly few genes. It was always assumed we had many more than the 22,000 we now have, and so the total blueprint is 3 billion letter units. These encode only 22 or so thousand protein coding genes, and this makes up only 1.5% of the genome. A lot of that extra coding, these extra letters, actually play a critical role in turning on specific genes in specific times and places. And what we've come to realize now is that a lot of the complexity of being human comes from how and where we turn genes on and off based on this instruction, rather than the complexity of the genes themselves. I'll point out to you that the 22,000 genes that we carry, this is Jim Watson, who helped decipher the structure of DNA, the 22,000 genes that we carry are less than grapes. You can interpret that however you wish. Now, one way of looking at these 22,000 genes is as the building blocks of life, the 22,000 proteins, each expressed in their own place and time, making us who we are. This can be viewed in some ways as the periodic tables. So every molecule is made up of one of the elements on the periodic table. They're the basic building blocks of chemistry. And in an analogous way, these 22,000 genes are the building blocks of each of us. They determine, to a large extent, not entirely so, but to a large extent, who we are and what we're like. Now, there's a major difference between these genes, however, and the elements in the periodic table, and that is these elements are immutable. Nickel is nickel. Now, you can have isotopes of these elements like nickel, but by and large, nickel is nickel. It'll behave similarly in most cases. The genes, the 22,000 genes that comprise us, are not immutable in the same way. So of these 3 billion subunits, some of which of these are variable from person to person. So each of our 22,000 genes is not identical to our neighbors. In fact, on average, 1 in 1,000 bases differs from one person to the next. This corresponds to about 10 possible differences per gene. And this means that between any two of us, there are 1 million total spelling differences between us, 1 million instances in which the blueprint is slightly different. And it's these differences that account for our unique characteristics. It's as if you were reading a sentence and instead of saying, I am the gene, you said, I am the gyne. This would be a spelling error, and the analogy here would be if instead of normally reading ATAG, you would read ATCG, this would now encode for a different amino acid that in many cases has consequences. Okay, so there's spelling differences in the blueprint in the book that comprises all of us. Now, there's a pretty simple way to quantitate the extent to which these spelling differences among all of us contribute to genetic differences or phenotypic differences, different characteristics among us, and that's by looking at identical twins. This is actually a family of two identical brothers who married two identical sisters and had non-identical kids, but we could talk about that. It illustrates a lot of how early genetics was done. It's because identical twins share genes and environment, or all their genes and environment, they come from a single egg. Fraternal twins, by contrast, share fewer genes and still share their environment. So what happens is, if identical twins are more similar for a given set of traits than are fraternal twins, you can quantitate the genetic contribution to that trait. And there are some astonishing things that emerge as having a genetic basis from analyses of this sort. I'll show you some of the numbers in a moment, but here are some other things that genetics may in time explain. I'll just let you digest this for a moment. I'm not sure who put junk in the trunk in, but it wasn't me. Well, here's the quantitative data for a number of traits. I study obesity. It turns out obesity is highly heritable, more heritable than any trait that's been studied besides height. We're trying to understand the genetic basis of obesity, but others are studying the genetic basis of schizophrenia, diabetes, hypertension, alcoholism, epilepsy, heart disease, breast cancer, and so on. Now, what data of this sort means, and we'll come back to this, is that none of the heritabilities in any case are 100%. So none of these things guarantee that you'll be obese if that obesity, for example, is 100% genetic. There are other contributors, as there are for these other conditions. What this does, however, tell us is that genes contribute, and what also is the case is that we can now begin with increasing rapidity and greater precision, identify the genes that contribute to these and many, many other disorders. Now, beginning in the 80s, when I started my work and other people did similar work, a set of genes were evaluated because they were more tractable for analysis of the sort that will allow you to read out the spelling difference. And so over the years, single genes disorders had already been identified for conditions such as muscular dystrophy, Huntington's disease, cystic fibrosis, hereditary forms of cancer, breast and colon cancer, hypertension, diabetes, and, as I mentioned, obesity. In fact, in our case, our work led to the identification of a genetic mutation or spelling error in this boy that led him to be obese as a result of a defect in a single gene that encodes a hormone that regulates body weight. So in this child, a single spelling error in a single one of those 2,000 genes disrupts the expression of a hormone that we named leptin, and if you treat this child with the leptin that he's missing because of a spelling error, this is what he looks like 5 years later. So the identification of single gene disorders, which has been possible since the 1980s, has informed a great deal about a great number of human diseases. And these are cataloged in databases that are available to all of us. What's shown here are some of the data that you could glean from these databases. The breast cancer gene, identified in part based on the work of Mary Claire King, allows individuals with a family history to find the spelling differences that lead to this predisposition and take appropriate steps. The same is true for colon cancer. The cystic fibrosis gene was identified, the spelling error is known, and couples who want to have children can screen for this periconception. Women who have patients who have factor V mutations can be susceptible to thrombosis, and because of this risk factor, there are clear medical consequences in the form of not taking birth control pills and so on. And some patients have spelling errors that lead them to synthesize less effective drug metabolizing enzymes, which is really important if you're going to be taking drugs to know that you won't metabolize it properly. Now, this is the starting point. Thousands of single gene disorders have been identified, but it doesn't explain everything we would want to know about our disease risk and how we should manage our health optimally. And what's driving this change, the change I hope we have a chance to talk about at some length tonight, is that it is now possible to read out the entire blueprint all at once. You can read out all 3 billion of your base pairs and define what your specific blueprint looks like for an ever-decreasing amount of money. In fact, the cost of reading out this blueprint for each of us is falling faster than Moore's law. This is what Moore's law would predict, and look at how this has fallen. The current cost to sequence one's genome is $3,500. It's expected it will come down to $1,000 in very short order and keep going down. So I think in our children's lifetime, you'll be able to sequence your genome for $100. This means everybody is going to have their genome sequenced. And that's the world that we're going to be living in. That's the world our children will be living in. And in many ways, it's a different world than the one we inhabit today. Let me give you an example. You could read out the entire blueprint. These are the 4 bases, the letters, as it were, of the alphabet that spells out our protein sequence, and look and say, oh my goodness, these 3 bases are missing. That's the cystic fibrosis mutation. Well, as I've been alluding to, I believe this is going to have a major impact. Making predictions is risky, particularly about the future, as you all know. But I'm really going to go out on a limb here and share my view and the view of others that genome sequencing will be at the center of 21st century medicine. And let me extend this to some what actually don't appear to me at least to be very risky predictions. Understanding the molecular basis of disease in the way we'll be able to by reading out the blueprint will make it possible to identify people with predispositions before a disease develops so that you can make adjustments if you need to. Therapeutics will be developed that target the specific causes of a disease, the particular spelling error that leads to the disease, rather than lumping people together because they have diabetes. Different people likely have diabetes for different reasons. We'll be able to decipher that by being able to read out the genetic code, and this will lead to an era of personalized or precision medicine. One critical point I'm going to make now and make again later, however, is that these spelling differences don't necessarily predict with absolute certainty an outcome. They reveal probabilities of an outcome, giving us an opportunity perhaps to make adjustments, as I just mentioned. And finally, there are many medical, social, and ethical challenges associated with this new knowledge, and I'll briefly review some of them. So I want to quickly now run through a few examples of each of these 4 bullet points. Let's talk for a moment about Alzheimer's disease. There are differences in your genome that could very precisely predict your likelihood of developing Alzheimer's. Some of these are pretty catastrophic and lead to early onset Alzheimer's, but other changes, in part based on the work of Jan Breslow, one of my colleagues here on campus, identified a gene known as ApoE, and variation or spelling errors or differences in the ApoE gene have a dramatic impact on the likelihood that patients will develop Alzheimer's disease. This information is also being extended to cancer. All tumor genomes in one point, all tumor cells will be sequenced at a certain point in time when the cost falls. And this allows us to subtype cancer. Again, people may lump cancers together based on the site at which they first develop, but that doesn't mean they're the same type of cancer. So we'll be able to subtype cancer based on the sequence, and this information combined with clinical data will help us pinpoint specific tumor cell types and identify those specific types that respond best to specific drugs. This also will allow us to identify patients who may be helped or may need experimental drugs, and this recipe of better drug targets, specific information about specific tumors, and improved patient selection will lead to more precise medicine. Gene variation of this sort is also important for the response to drugs. I'm sure many of you are aware of the fact that adverse reactions to prescription and over-the-counter medicines represent a significant health risk. And to give you an example of how critical this can be, I want to tell you of another instance for the treatment of childhood leukemia. In children with this disorder are often treated with anti-cancer agents, some of which are listed here, azathioprine and 6-mercaptopurine, and this enzyme degrades these drugs. Well, some children have abnormalities in the enzyme that degrades these drugs, and if you have a spelling error in the enzyme that degrades this drug, you can have a hypersensitivity to these drugs and actually die. So you can have a lethal consequence to giving the normal dose of this drug to children. And so TPMT testing for the spelling error that alters metabolism of drugs is critical for patients who receive them, and here are some of the indications for those drugs. So just about every aspect of medicine will be touched by an ever-increasing database of spelling differences and their phenotypic or clinical consequences. So what are some of the challenges associated with the ability now to read out your DNA? Well, as I mentioned, what you'll get, and I suspect those of you who availed yourself of the service of 23andMe saw, was that you don't get a guarantee that you're going to develop a problem. What you get is a probability. And this is both, for some people, I suspect, a crisis and an opportunity. The crisis is you may see things you didn't want to see. On the other hand, you may be able to intervene earlier than you would have otherwise. There are mutations, for example, that could predict very precisely that you're at higher risk for alcoholic liver disease. If you have that mutation, then you may be able to adjust your behavior in a way that reduces your risk. And pretty much everything you look at will be probabilistic and not a guarantee. So we have to get used to looking at information in a way that doesn't say, oh, I have this is my future. We're going to look at information and say, well, this increases my risk for this and that, and as a consequence, there may be things I need to do now to reduce that risk. There are other things that come of this, and that is that there are no perfect specimens. Everyone has a probabilistic risk of some disorder or another, and it will be up to us to help to try to manage this information and use it wisely. This information will also exceed what any one person can keep track of, and so there will be computational tools for managing these data that are going to be essential for making optimal use of it. And I think actually because of this, patients are going to become increasingly active participants in the process. A doctor isn't going to look over your genome sequence with the enthusiasm that you might because he has many other patients. So I think it'll be up to us to begin to try to understand what this information means and doesn't mean and chart our life's course accordingly. The day will come, in fact, where we won't actually even have this information, I expect, on thumb drive. It'll actually get implanted on chips. So everybody will have available to any physician who needs to access it their complete blueprint with the readout of the DNA sequence and the protein sequence of all the genes all 22,000 genes. What does the world look like then? Well, actually, you may have a reaction to this. It looks a little bit like Gattaca, the movie Gattaca. Did anyone see this movie? Well, it sort of illustrates the fact that you can use the information that will be gleaned wisely and ethically or not. And I think it's important that we all think about this so that as the time comes and as the time comes for our children, we do use it wisely and ethically. So to illustrate some of the challenges that will be associated with this, I want to ask you to think about the following questions. If you were reading out the blueprint of your children, and this is already possible for children in utero, so you're reading out the blueprint of your unborn child, ask yourself, what would you do if someone told you, a doctor told you, that your unborn child was at risk for a treatable disease, such as phenylketonuria, which can be treated with the diet? Would you do anything? What would you do if you found out you or your unborn child had a 50% likelihood of developing a serious neurologic disorder such as Huntington's disease, which is what Woody Guthrie died of? What would you do if you or your children had an increased risk of developing Alzheimer's prematurely or heart disease? Would you do anything or not? What would you do if the blueprint for your child said he was likely to become obese? Or had an increased likelihood of becoming obese? I was actually asked, I was blindsided with a question about this on a talk show once because they told me that 40% of patients polled, according to them at least, would abort a child if they knew the child was going to be obese. I'm really glad my parents didn't have that test. So which of these would you want to know, and what would you not want to know? Do you want this information? I think the axiom in medicine is generally that you want information if it's actionable, if there's something you can do about it. But many people don't want to know if there's nothing they could do about it. This equation changes, however, if we're thinking about an unborn child and one's ethics associated with that as compared to yourself. So which would you want to know? And importantly, what should anyone else be allowed to know? Should someone else, any agency, an insurance company or anyone else, be allowed to know what your risk or your family members' risks of disease are? Now, this is a serious issue, and it's one being taken seriously by the government. There's legislation to ensure that genetic information remains private so individuals won't be discriminated against or deemed uninsurable based only on their DNA. But I want to close by telling you that it may not be so simple as to simply proclaim this, because there could also be cases where patient privacy would conflict with the public good. I think we all endorse the idea of privacy as an instinct, but let me tell you this one story that reveals that life can be a little more complicated. And it's about one of these thousands of genetic disorders known as long QT syndrome. Long QT syndrome is a rare hereditary disorder where the heart's electrical rhythm can become deranged. And it leads to circumstances in which otherwise healthy, normal people can develop a very significant arrhythmia known as torsade de pointes. It's a very rapid rhythm, and patients who have this will suddenly one day for the first time faint with what can be oftentimes a fatal condition. Sudden, unpredictable. There are 7 different mutations or spelling errors that will cause this disease that have been discovered so far, and all encode an ion channel that functions in the heart that has been under intense study the class has been under intense study here by Rod McKinnon, who actually won the Nobel Prize for studying this class of proteins. So mutations in these genes can cause sudden arrhythmias and fainting and even sudden death. And this disease, or this spectrum of diseases, can be identified by genetic testing. So let's think for a moment. Would an employer or the public have the right to know that someone had long QT syndrome under any circumstances? Does the public have an interest in knowing this information? Would the public have a right to know, for example, if their stockbroker had LQTS? Well, definitely yes. No. A CEO of a company that's publicly traded? Probably not. What about a bus driver or a school bus driver? Would you have a right to know that your bus driver was at risk for fainting suddenly if they were driving your children? What about an airline pilot? Do we need to know that an airline pilot is at risk for this disorder? And what do we do when we sequence the genome of an airline pilot and you now see the spelling error that tells you this person is predisposed to it, to this sudden death, potential sudden death syndrome? And it can get even more complicated, because what would we do if the pilot didn't have his genome sequenced? His brother did. And his brother, it turned out, or sister, was found to have the mutation that can cause this potentially lethal condition. Well, now we know that the pilot has a 50% risk of having the condition. What are we going to do about this information? Now, actually, I'm a scientist, I'm a geneticist, I think about this all the time, but I want to be clear, this isn't my decision. This isn't geneticist decisions. This isn't scientist decision. This is all of our decision. These are the sorts of challenges we're all going to face, and collectively we need to get it right, because we need to be fair and we need to preserve individual rights, but we also need to preserve the public good. And I'm not in any way suggesting where the line should be drawn, but the question is already upon us. All right, I'm going to close now and just tell you where I think genome sequencing is and will go. It's going to have a big impact on healthcare. It's going to allow us to define the genetic and also provide the basis for understanding more about how environmental factors contribute to disease. It's going to lead to an era in which our blueprint will allow us to develop individualized preventive medicine. We'll be able to develop new therapeutics based on the root cause of disease. This is going to require, however, that the public and health professionals be educated and have the necessary tools to use the information. It's going to create, actually, healthcare disparities, as you can imagine, and create privacy issues that we all need to think about together. This is going to be the world in which my children live. It's going to be the world in which their children live going forward. And I think dealing with this revolution that's upon us is going to require that we all be scientifically literate, we be able to navigate our own healthcare challenges, but also think about the collective the challenges that we'll collectively face. So I want to thank you again for visiting our institution. We're very proud to be associated with an institution with a record of distinction of the sort that this one has. And so I want to thank you for visiting tonight, and I think there may be time for some questions if any of you have any.
Well, thank you very much, Jeff. We do have about 20 minutes for questions.
Outstanding lecture, Jeff. What I'd like to ask is what sort of techniques are being evaluated and tested that have a potential of correcting spelling defects, and how much progress is being made in this area with the human genome in this area?
Yeah. So I think there are lots of challenges that are going to make it very difficult to actually go in and fix the spelling error, as it were. So I think it would be a long way off till you could say, well, this person has an alteration in the gene, and we're going to fix the gene itself. I think it's more a matter that the knowledge of the fact that it's this gene that's contributing to this condition will give us the intellectual framework with which to now develop new treatment. So, as an example, for the boy who's genetically obese, I don't think it's foreseeable, at least to me, that you could go in and fix the spelling error so that his gene would now make the protein that's lacking. On the other hand, you can make the protein he's lacking in the laboratory and give it back to him. So that's a simple example of how the knowledge can lead to new treatments. And there are many other examples of that sort.
Thank you for that. I'm just curious, you talk about this lecture as being about our children, and I'm obviously the techniques for understanding the genome are here today. What about the cures? And in your view, what is the timeline for when these two things are going to meet?
Yeah, so the amount of information that's being generated is staggering. I can't remember I think, in fact, some people say the limiting factor is quickly becoming how much information you can store and transport. There's too much information, for example, to put over the web anymore, and people sometimes have to ship boxes from here to there, with hard drives and so on, to transfer the information. The point I'm making is that we're reading out the spelling errors, or differences, could be either, much faster than we're able to figure out which differences correlate with which diseases. So the knowledge is accretive, and I think we're associating particular spelling differences with particular outcomes with increasing frequency. But this is going to be a process that's going to take decades. So where are the cures going to come from? I think it's a case-by-case instance. I think, in some instances, this new knowledge will lead to obvious next steps. In other cases, it may take more time. And I think the transition from the new information to actionable information that changes disease treatment is going to be more relevant for our children than it is for us.
So there's no hope for us?
There's no hope. It depends what you're hoping for.
Thank you very much. I'm curious about identical twins. Have there been genetic sequencing to determine whether the environmental effects during the course of their lifetime has changed the genes that they have? And so, what does that tell us about our environment as well?
So you'll note from the heritability estimates, which the question refers to, the fact that it's never 100%. So something other than the genetic sequence is contributing to differences among twins, because no two twins are or very few are completely identical. So where do these differences come from? Well, one difference could be environmental factors or sets of experiences that influence them differentially. There's another cause of variability, though, that's getting attention and that will also be revealed by this technology, which is something called mosaicism. We all start out as a single cell, and the cell divides, and the cell divides again, and the cell divides again, and out of that single cell that came from our parents, from zygotes that were derived from our parents, we come. Now, oftentimes when the cells divide early in development, you can accrete spelling errors then. So maybe when you went from 2 cells to 4, one of the 4 cells had a spelling difference that now impacted all cells that were dividing subsequent to that one, or that were downstream of that one. That's called mosaicism because it means that the person who has that spelling difference is a mosaic of 2 different cell types, one that had a spelling difference from all the rest. And so there's actually a lot of DNA sequencing being done to compare different tissues from identical twins to actually catalog what spelling errors have now accumulated in specific tissues based on these mutations or spelling errors that occurred during development. It's actually a really interesting opportunity because it may provide a quasi-genetic explanation for differences among twins or between twins at the same time as we can also think about what environmental factors may be relevant.
You show us a few conditions that depend on one gene abnormality. Are the majority of the medical conditions due to multiple gene abnormalities, and how complex does it get there? Then?
Yeah, Mark, how many? I think there are thought to be 4,000 single gene disorders, something like that. So there are 4,000 cases of human disease as a result of what you're referring to, which is a single spelling difference in a single gene. But most diseases are much more complicated than that, and things like hypertension or heart disease or obesity for the general population, not the child I showed a picture of, are likely to be a result of a combination of genetic differences in a compilation of genes or multiple genes. And that's one of the challenges that we face, is that it's going to require incredibly complicated mathematical tools now to be able to say it's this combination of a few genes that contribute to what are referred to as complex diseases. Now, what I alluded to in the talk, but I didn't really go into, is this. In the 80s, it was possible to identify the defective genes for single gene disorders like Huntington's disease, cystic fibrosis, the form of obesity that child developed, and so on. And that was the low-hanging fruit, as it were. But nowadays, with the abundance of information for an ever-increasing number of people, the notion is that we'll be able to put together specific combinations of genetic differences that contribute to more complex diseases.
Thank you. I think I heard you allude to junk DNA not necessarily being junk. And what discoveries or implications are there from that affect this context?
Well, so I actually don't particularly like the term junk DNA because it presupposes we know that it doesn't do anything, and we don't know that the DNA in between the genes doesn't do something or many things that are important. It's clearly part of those DNA sequences in between genes turn them on and off in specific times and places. There are specific functions for other DNA sequences in there. But I think there's a growing humility about our lack of knowledge about how all the 3 billion base pairs might function and play specific roles in development. The idea of junk DNA comes from a concept, a very clever concept by Richard Dawkins, who you might have heard of, who pointed out that if DNA found its way into your genome and it could replicate and didn't do any harm, it would stay there. And he referred to that as selfish DNA. So clearly some of the DNA in our genome is selfish DNA and probably without a function other than propagating itself in a selective way. But I think there's a lot to be learned about these intergenic sequences, a lot of attention being paid to it. It's much harder to study, however, so I'm not sure how quickly we'll make the sort of breakthroughs we'd like to there.
Is DNA static, or if I had two 23andMe tests some years apart, would there be any differences?
I think from test to test in an adult, it ought to be pretty similar. It might depend a little bit on where the DNA came from, for the reason I mentioned of mosaicism. But I think DNA is designed by evolution to be pretty static and pretty stable. Errors get made sometimes when it's copied, but the DNA itself in non-dividing tissues is pretty stable.
Have you had your DNA sequenced, and would you recommend that one have their DNA sequenced?
I have not. I don't have any particular desire to, because I'm not sure how well I would cope with the glitches I would find. I feel like I know enough already. Would I? No, I don't have any desire to. I guess what was going through my mind is, would I encourage my kids to have their DNA sequenced? I probably should, but we haven't done it yet. But when the cost comes down, I think at a certain point in time, you probably need to take your head out of the sand and find out what you need to find out to make the right decisions for yourself.
So if you did a study of 1,000 or 100,000 individuals, what percentage of them would have a defect in the sequence of their DNA?
Every one of them. It depends how you define defect, of course, but every person has DNA sequence variation that would be considered less desirable than perhaps others. And
let's say potentially a very serious defect.
Well, I think for most of them, they're going to be probabilistic. The only ones that I think in the lexicon you're using that would be very serious would be a single gene disorder, something that totally disrupts a gene, so you don't make the protein. And these things you could say are pretty serious. I listed some of those before. But most of these spelling differences don't completely disrupt the gene altogether. They change its function in subtle ways. So I think everybody would have DNA differences that you could probably consider less desirable that subtly change the function. But I don't think necessarily you'd have something that you could say with certainty is going to lead to a disease, as you could for the child, for example, who has the defect in the leptin gene, where you could say with absolute certainty this child will become obese. Does that answer your question?
Not really.
Should I try again or take it up over
No,
to
it just, you know, you've suggested that maybe we should think about having this sequence done, and yet, you know, without knowing the probability of how bad the news would be, and that you have not decided to do it. I'm just curious, probability-wise, what percentage of us would be seriously disappointed in
Well, Mark, you had your sequence done. What was your reaction?
So I'm in the opposite camp. I absolutely want to know my sequence, and I was very interested to find out, certain higher risk factors. Some of them that really rang true because they related to conditions in my family that I knew a grandmother had passed away from a certain disease. And so, it makes me more alert to that. Again, it's just probabilities that you see for the most part, unless you have some of these causal single gene mutations where if you have it, you definitely get the disease. And actually, I do have one of those. I have hemochromatosis, which, high blood iron. That came out of that analysis as well. So I think it's incredibly useful, and I certainly, for one, wanted to know, and it's been very valuable for me to have that information. I'd like my kids to do it. They're less interested in doing it, I might add.
I'm realizing that I may not have put myself in the most desirable position by arguing how transformative this is and then telling you I don't want to have it done. But in fact, we might have been able to figure that out from my code. Doesn't want to know. Gene 1143, C to T.
Thank you so much for that lecture. It seems to me that the rate at which knowledge has accumulated since that, I think it was 1928, is just absolutely incredible. And could you forecast that rate of knowledge accumulation will continue? If we were here 10 years from tonight to hear a lecture like this, would there be dimensions more that would be discussed, or are we getting near the top of this particular subject?
Well, I think in a way the answer to that question depends on whether you're asking, are there great conceptual leaps left to be made about how we think about ourselves in the world? And I resist very much the idea that there are no conceptual leaps left to be made. And if anyone can tell me what they are, I would gladly take them into my laboratory. And in a sense, that's what we dream of, is trying to advance our understanding. But you either see it or you don't, and I don't see any of the sorts of things that would qualify at the moment, but they'll come. That's just progress and the march of knowledge. What I can say with certainty, however, is that there'll be very important details being added with greater and greater frequency, and these will affect people's perception of their health and the way they'll want to manage their futures to some extent. So, for example, if you knew you had hemochromatosis, I think probably alcohol consumption is probably not.
No, it's a very treatable disease.
Yeah. I didn't mean that the way it came out. I was actually, what I was thinking about is actually there's a condition known as alpha-1 antitrypsin deficiency, which I wasn't thinking of, and that's an enzyme deficiency that can cause cirrhosis. And if you have that, you definitely want to be drinking. If you have hemochromatosis, you should be drinking, is my understanding. Because you're worried about your iron.
Following up a little bit on John's question, given the rapidity of change and what's been done in the last few years, how is this going to change the nature of science, how it's done? And could you also comment on how you think we need to think about teaching science?
Yeah, I mean, I think the ability to use large data sets of this sort is going to be increasingly important in lots of different ways. There are other dimensions to this data that I didn't mention, such as comparing the DNA sequence of different animal species, looking at the inventory, making generating hypotheses based simply on looking at data. I'm not an expert in that area, and frankly, I'm not particularly, it's just not the way I think about things, and I wish I were better at it. But I think it does add a dimension in terms of how you go about your business, because if you find some observation or some data, you need to go to the database. You need to look at what sequence variation is available for that gene, what its impact is, whether other animals have that sequence variation and what impact it is, and so on. So I think being able to access and better yet, use information is going to be a critical component of biomedical science.
Jeff, that was terrific. Could you talk a little bit about leptin for everyone? What did the discovery of this material, what does it lead to in terms of translational science that we can all identify with and relate to, and what will it do?
Well,
that's the layup question.
Yeah, actually, that's a pretty, it's like, it would have been probably better if you asked me what my favorite color was or something, but like from Monty Python. Green, I mean blue. How could I, well, you really crossed me up with that one, Alan. Well, I guess this sort of gets back to the question David asked in a way, which is that sometimes finding a gene reveals some things you didn't have any idea existed before. I think it's fair to say that the conventional wisdom before our laboratory identified leptin was that body weight was not tightly regulated. There wasn't necessarily a physiologic system that controlled weight. The identification of this gene and the realization that there's a hormone made by fat tissue that essentially reports nutritional information to your brain as part of a biological system that regulates weight was an advance. And it's an example in a more general sense of the fact that sometimes you find a gene that causes a disease when defective, and it leads you into a whole new pathway that changes lots of things. Another example of this is a disorder that some of you may have heard of called familial hypercholesterolemia. This is a defect in a gene that plays an important role in cholesterol metabolism. When it's defective, children die of heart disease sometimes before they're 10. Brown and Goldstein, who won the Nobel Prize for this work, and Joe Goldstein's on our board, identified the defective gene. The disorder isn't very common, but the realization that this gene plays a critical role in cholesterol metabolism in part paved the way for statins, which I suspect many of you take. So sometimes, and I should take, but don't for the same reason as I don't want to know lots of other things. But, anyway, the basic idea here is that this new knowledge leads you into a pathway where other tools can really reveal biological systems and control points that you didn't know of before. And I'd like to think that leptin has had an impact of that sort for our understanding how weight is controlled.
Yes, thank you. You've talked about the differences in predisposition toward diseases, but how far can you take this? Can you talk about predisposition toward scientific literacy and intelligence or personality or other? So how far can you investigate the genome towards other human traits?
Well, that's a really good question, and in a say that's a set of ethical questions I hadn't thought of, but that could have easily included. I suspect there are genetic differences that account for not only the physical characteristics we've been talking about, but cognitive characteristics as well. We can sort of see that in our kids. You can see that in identical twins. Clearly, genetics has a lot to do with not just brain development, but brain function and certain attributes and so on. I think that will be learned over time, and I think, as you're maybe alluding to, could create a whole other set of ethical challenges. What would you do, for example, if, it occurs to me, if a child had a particular difference that influenced the intellect in one way or another? Would you change their educational opportunities or not? You know, in some ways it sounds like it's a little bit like Brave New World. Not that we're going to be breeding people per se, but where you get information that predicts things with a probability but not a guarantee. How do you use that information? Should you use that information? I'm not sure where the lines get drawn exactly, at least as pertains to a disorder like long QT syndrome, but I think there'll be other analogous situations as more and more information is accrued.
Thank you again, Jeff.